Good evening, everyone, and welcome to the first of this year's Oxford Botanic Garden and Arboretum Autumn Science Lectures, sponsored by Plants, People, Planet. And this year, the lectures have a theme of plants and medicine. My name is Simon Hiscock, and I'm the director of the Oxford Botanic Garden and Arboretum, in case you haven't been to any of these lectures before. And as always, it's my great pleasure to introduce the evening's speaker, but only after I've done a bit of preliminary housekeeping information pass on. So firstly, this lecture is being recorded, but your camera and mic are turned off, so you are not being recorded. If you would like to ask a question to the speaker, then please use the Q&A function, not the chat function, and I will accumulate the questions and then ask them at the end of the talk. If you need to contact us on anything else, particularly sort of technical issues, then that's when you need to use the chat function. One of the team will get back to you quickly. The recording of the lecture will be available to all attendees after the event, and we will be sharing details of how to access it via the emails that you registered with. So without further ado, I'm now delighted to welcome our first speaker, Professor Julie Hawkins from the School of Biological Sciences at the University of Reading, where she is Professor of Plant Systematics and Evolution and Deputy Head of School. She is also an editor of Plants, People, Planet, which is the sponsors of these lectures. Julie studied for a DPhil here at Oxford in the Department of Plant Sciences, researching the systematics of a group of dry forest trees from Central America and Mexico. She then moved to a research fellowship at the University of Cape Town, where she studied the genus Aspelanthus, including Aspelanthus linearis, which gives us rooibos tea, otherwise known as red bush tea, and is a very wonderful tea if you've ever tried it. She later returned to the UK to take up a lectureship in plant taxonomy and systematics at the University of Reading. Her research there has included taxonomic studies of the bean family Fabaceae and the Cactaceae, and the ecology and diversity of the Cape floristic region of South Africa. However, her research over the last 18 years has placed a focus on medicinal plants, especially those used by indigenous peoples. In this research, Julie developed innovative techniques in plant systematic analysis to explore the taxonomic distribution of known medicinal plants as a means to identify related species that may contain important pharmacologically active chemicals with the potential to become the drugs of the future. This research will feature in Julie's talk this evening, entitled People and Medicinal Plants, Evolutionary Approaches. I'll hand over to you now, Julie. Looking forward to your talk.
Fantastic. I'm here. I've managed to arrive in the meeting room. Hello, everyone. Thank you for inviting me. I'm just going to launch my presentation. Here we go. Excellent. Surprisingly, I am not seeing what I was expecting to see, so I'm just going to take a moment.
Hello.
Hello.
Should we try from the beginning again?
Yeah, let's start back again.
That worked last time, didn't it?
Stop sharing. Here. Sorry, that worked so smoothly in practicing.
I know, don't worry. There we go. Start again.
Right, good. Okay, share screen.
Yeah. Then your
Use the
PowerPoint.
PowerPoint. Fantastic. Share that. Start the presentation. And we're off.
There we go. I'll let you carry on. Thanks, Julie.
Thank you. Marvellous. Sorry about that shaky start. We're here and ready to get going. Thanks again, Simon, for inviting me to speak today. Very happy to do that. You started us right at the beginning, my doctorate in Oxford. That was way back in 1992. And as Simon explained, I was very much being trained, learning the tools of the taxonomist, the systematist. That is the discovery and naming of species, understanding the relatedness of species using what we now call phylogenetic methods, and organizing a classification to reflect relatedness. And at the time that I was working on my PhD, this publication came out, Systematics Agenda 2000. I think it came out in 1994, so I was a couple of years in. And this was a document that my peers and I spent a lot of time reading, looking at, discussing. It was it had a big impact, I think, on people's perceptions of the importance of organizing and naming plants and other living organisms, and it was interesting as well because it put a lot of emphasis, as you'll see, mission 3, that the emphasis there was on making tools that allowed people to better meet the needs of science and society because of the work of systematists. And I spent a lot of time looking at this document, talking about it with my peers, and there are a couple of things in it that I remember rather well that were about medicinal plants. And one of them was introducing the idea of using I've got to put my phone down because I'm being distracted. Using medicinal knowledge of important plants that could yield important molecules for therapeutic uses, and finding those in close relatives. And the example was the example of Taxol, which is of course a really well-known example in medicinal plant studies. Taxol being a drug developed following the work of many years. So in 1958, the USDA screened 35,000 plant species, including Pacific yew. They discovered a active ingredient with promise as a cancer drug. Phase 1 clinical trials started, but at that time, the species from which the molecule was isolated, the Pacific yew, could only yield 0.5 gram from a whole tree. So the search was on to find another source for that molecule. And indeed, it was found that English yew was a much more, had much higher amounts of the molecule of interest, and it was developed as a source for further testing of Taxol and ultimately came to market. And I remember reading about this in the Systematics Agenda 2000 documents and thinking they are looking for the best example that they can find of how systematics serves drug discovery and has a predictive value. And there's absolutely, of course, Taxol is terrifically important, and that goes without saying perhaps, but what's interesting is that the predictive element of finding an alternate species that could be the source of a valuable molecule was a bit underwhelming. So they knew that Taxus brevifolia was a source, and the place that they looked was the English yew Taxus baccata. And I remember at the time thinking that's a slightly underwhelming example of how systematics can be predictive. So hold that thought. How can systematics be predictive? Here we've got if you like, the textbook example of looking at a species that's in the same genus as a species that's known to be a source of a valuable molecule. And another case study that was in the Systematics Agenda 2000 documentation was an example of the Madagascan periwinkle, another famous example, famous then because this plant yields a small molecule which has had a tremendous positive impact as a therapeutic agent for treating childhood leukemia. And again, you read closely into the example that the Systematics Agenda 2000 team put together to promote systematics in the service of medicinal plant research. And they're highlighting a plant which did have some folk use, but for actually a completely different therapeutic use, a different therapeutic application. The folk use of the Madagascan periwinkle was in diabetes, and ultimately small molecules from this plant were found to have value in treating leukemia. And so that's something that sat with me as well at that time. And I thought, okay, so here's some possible predictive value of ethnobiology, of the way that people in different communities in different parts of the world use drugs, use plants to meet their health needs. But how is this the best example that we have of that? And that was quite interesting to me. And also that Systematics Agenda 2000 highlighted that the reason we might use ethnobiology as a way of finding useful medicinal plants, useful new small molecules, is because these plants have been shown to be efficacious through what can be considered a long-term trial. That is, that the people who've been using those plants for a long time have effectively sifted out and identified those ones that are efficacious, and therefore looking at plants in this way is going to open up the possibility of finding more and novel small molecules that can have therapeutic use. And indeed, we can see where these ideas came from that were embedded in Systematics Agenda 2000. The idea that ethnomedicinal use is going to, or the use by communities who've tested the plants through time, is going to yield useful small molecules. It's heavily promoted by Norman Farnsworth and his work. This is an important paper, cited a huge amount. It's from 2001, and he presented a table of the compounds from plant species showing how the way that people in different cultures are using them in what he calls ethnomedicinal use maps to how the derived drugs are used. And he also highlighted how little we know. That's 6% of plants are known pharmacologically, 15% phytochemically. So this was the sort of background where Systematics Agenda 2000 was building on, and this idea that that prior testing and a long-term trial and error is how we might infer efficacy of the plants that are used traditionally. This is an idea that we can trace back to Richard Schultes, and this is him here on the left, and he's 21 in this picture. He's not going to go on to be one of the most famous ethnobotanists of his time and to set the agenda for the next sort of 40 or 50 years of ethnobotanical research. This is him after a a peyote ceremony. He's with a student colleague. He's 21. He's doing his undergraduate dissertation in this photograph. His thesis was a study of the ethnobotany of peyote, hallucinogenic cactus. Apparently, as part of his undergraduate thesis work, he visited 15 indigenous groups and ingested himself peyote 2 to 3 times a week. I was just doing some health and safety reviews for some student projects, and so it's extraordinary that this was his student work. But he went on to, as I say, to be a huge advocate for the work of ethnobotanists, and specifically interested in motivating for the collection of traditional knowledge of medicinal plants, realizing and arguing that this was knowledge that had been developed by generations of testing, in place, and highly conserved, he argued, but also currently under threat. So he influenced a generation of ethnobotanists to collect rich data about how medicinal plants were used by many people around the world. So this idea of predictivity based on relatedness of plants, and also based on the way that plants have been used, was something that's captured at the time I was doing my PhD all those years ago. And I finished my PhD, I went off, I did something else, I was doing systematics, taxonomy, but so many students came to me, and I had to find student projects for a number of students every year, would come to me and say, I really want to study medicinal plants. And I wasn't uninterested in medicinal plants, but I did not want to supervise a lot of phytochemistry projects. So I started to think back to this idea of predictivity that I'd come across as a student, and thinking about what kind of projects could I offer them that would capture some of these ideas about predictivity. And at that time, I came across the work of Daniel Moorman. And this is work from Daniel Moorman from, I think, shortly after the publication of Systematics Agenda 2000, so sort of towards the end of the 90s. And he had brought a new way of looking to how people choose plants. People of different cultures have created a group of plants which you might call their medicinal flora. And he had thought that what would be an interesting way of considering all of the plants together that are used would be to look at the overused and underused plants. So here we've got a plot of the number of species in a plant family against the number of medicinal species in the family. And what he showed was that you could find plants that were used more than were expected in light of the number of species and their availability, and plants that were used less than expected. And here we see Asteraceae as an overused family, Poaceae as an underused family. And he supposed that we might find similar patterns of overuse and underuse in different cultures if we were to compare the plants used by the peoples of North America with the plants used by the peoples of, he actually did this, of Korea and other countries. We might find similar patterns. And he started to think about why that might be. So there I am, 10 years after my PhD in Reading, finding student projects. And I think this is going to be a really cool thing for students to do, because they can find published data, and we can start collecting data about these patterns that you find in different cultures in the way that plants are used. And I have to say, at that point, I was hooked. And I have, from those early student projects, been developing these ideas about the diversity of ways in which people have made use of the plants around them and have selected the plants around them. There are probably 7,000 peoples in different ethno-linguistic language groups, depending on how you count them. But, you know, people are diverse. We have diverse peoples. And there's, you know, more than about 400,000 vascular plants. And so there's and following Schulte's work, there's been a generation of ethnobotanists, and even before that, of course, who have collected really rich data about how these plants are used. So they will collect information about the the local name of the plant, the part of the plant that's used, the way the plant is processed, what particular uses the plant is put to, what we would call the therapeutic applications. So that might be for stomachache or contraceptive use or whatever, a whole range of different therapeutic uses. And these data are dispersed through the literature and are still being collected. So here I've got, this is a student of mine in Morocco collecting plant use data with prior informed consent and a prior agreement to return data to the community in the form of a book in local language and French, so that it can be conserved by the community there. Another student of mine collecting the plant uses by women in Mecca for medicinal purposes. A student bringing together the data about the way that medicinal plants are used that are from publications and from annotated herbarium specimens, because a lot of herbarium specimens do record how these plants have been used. Bringing together is a huge database of diverse uses. And not to forget that pharmacopoeias, written pharmacopoeias, which are the sort of government guides of how the plants that are used medicinally and recognized by governments, also record a huge amount of data about how plants are used for different countries. We have the British Pharmacopoeia. We don't have a lot of medicinal plants in it, but many pharmacopoeias have a huge number of plants and traditional uses of plants recorded in their pharmacopoeias. So people are really diverse, and the ways that people have used the plants around them are also really diverse. And I became more interested in how we might explore the diversity of peoples and the diversity of plants together to ask questions about how plants are selected and what plants are selected, and how this might have changed through time and through space at different scales. That was a long introduction. We're on to the data and some of the work I've done. So this is one of my first studies, and what you see here is what's called a molecular phylogeny. So this is like an evolutionary tree of the relatedness of all the plants, in this case in South Africa, Nepal and New Zealand. And where things are joined together here at the tips, that means that they're closely related. And if you compare this plant to this plant, you see there's a lot of evolutionary distance between those. And here we extended this idea of Mormon, that you can find patterns of use cross-culturally, but in perhaps a slightly more sophisticated way, because instead of counting whether families were used more or less, what we did was we said, let's see if we can see whether the sets of plants, what you might call the medicinal floras, the group of plants that people are using, whether they're similar or different for different peoples. And we can measure that in an evolutionary tree without people having to use the same species or the same genera, because we can measure the distance between the plants that somebody uses in South Africa and how closely related that is to the plants people use in Nepal and the plants people use in New Zealand. And we can by doing that, we can actually have a much sort of finer grain than just by saying same family, not same family, because we measure everything that we can know about relatedness. And so we made this study. We knew about the relatedness of the plants. We had 3 groups of peoples. We had all of the information about the plants that they used. as far as had been published, and we found that they were predictive. So what people do in South Africa predicts what people do in Nepal, what people do in New Zealand, and so forth. So people are independently, because we chose places where we could expect little transmission of knowledge, little that people weren't telling each other about these plant uses. We inferred independent innovation, that people had independently come across groups of plants that were really important in medicine. And what's interesting about that is, for me, was how people reacted to that. Because on the one hand, you had a lot of ethnobotanists saying, well, yes, of course, because we know that through many, many generations of trial and testing, people discover the same species or related species because medicinal plants are efficacious. And on the other hand, we had this big group of people saying, that's quite extraordinary, because our expectation was that although there are no doubt many plants that have given us valuable small molecules that have become drugs, from our perspective, much of the plant use in a whole medicinal flora, across the whole medicinal flora, is going to have a magico-symbolic or meaning or work by a placebo effect. So we are surprised to find this level of cross-predictivity. So that was one study that showed us something about independent innovation. Next study I want to quickly tell you about looks at a situation where we've got many more different peoples. And this is a study that we did of the recorded plant use in Nepal. So Nepal is culturally a very diverse country, and there are people who belong to many language groups in Nepal. The two main language groups being the Tibeto-Burman language group, and these are examples of the languages spoken by people who speak Tibeto-Burman languages, so the Sherpa, the Tamang, and the Indo-European languages. And there has been a huge effort to record the medicinal floras of the peoples of Nepal, such that we can have a list of the plants used in medicine by the peoples belonging to each of these groups. And because we know how the languages are related, we can infer something about how long ago these peoples split from each other. If you think of humans diverging as their languages diverge, they diverge culturally. This is the thinking of evolutionary anthropologists. And we also know about their plants, and we know where they live, so we know what plants are around them. And these little maps show actually where people live. And so we wanted to test hypotheses about what was the predictor of which plants people use. And so this little phylogeny here, this is a made up phylogeny to reflect the relatedness of peoples, not plants. So the last slide I showed you, that was the relatedness of plants, and those were how plants were related, but actually people can be related as well. So we can have, for example, the Tamang and the Gurung people are more closely related to each other than either of those peoples are related to people who belong to Indo-European language groups. So one possibility in terms of how plants are selected is that the biggest driver of the selection of plants is going to be who you are, so what language group you belong to. So that would mean your group of plants are more similar if you are close relatives. Another hypothesis is that if you live in a different floristic region, you will make best use of all the plants around you. So if we think of this dark grey box as a floristic region, we can see that these peoples, these three peoples all belong in the, all live, in the same floristic environment. So they've got access to the same plants. And we might think, well, that is the biggest predictor of what plants they use. So this is, here we have a second hypothesis that these people, that the people in this floristic region have similar medicinal plants, and the people in this floristic region have similar medicinal plants. And our third possibility is how close you live to people speaking other languages is going to be the biggest determinant of which plants you use. And so that might be, here we've got people that, although they are in slightly different floristic environments, they have opportunity to exchange knowledge. And that might be a predictor of the plants they use. So this was very novel. I'm really proud of this piece of work because it allowed us to tease apart some questions about what it is that that might best explain the diversity of plants people use. And we were expecting that it would be who you are, your relatedness would best explain your medicinal plant selection, because of the work of Schultes and the narratives around Schultes that were, plant use is really deep in place and is passed on with a lot of fidelity because it's so important. People maintain knowledge. So it was very interesting to find that what predicted the plants that we used was actually the plants around you. So people, although they might have relatedness to people who use people were related to people using different plants and had adapted to a new floristic environment where they were able to select the plants around them. That was really interesting because it fitted with an emerging narrative that was challenging a little bit the ideas of Schultes and those who had followed him, that you had this strong in-place knowledge. And this is, Robert Works is a geographer who is really interested in what he calls the jungle medicine narrative. So he describes this idea of like a museum of traditional knowledge that is that is really strongly deep in time and place. He describes that as a jungle medicine narrative, as a myth that's really attractive to people. He argues that it doesn't reflect the way that people actually are very quick to bring new plants into their repertoire if they see them as being valuable. And he writes about this as a geographer, where he looks at case studies of a particular invasive plant. But it was interesting for us that we found this this pattern of adapting in our cross-cultural study, where we could test that against other possible drivers of similarity and difference. And just to flag here that we found something a little bit similar in this work that was done by a student of mine in Morocco, and you saw her earlier, Irene Texidor-Tornu, and she worked with women in Morocco who treat children, infants, and the women who do this kind of medicine, they're called Ferragat, and the kind of medicine is called Frig. And she interviewed a lot of elderly ladies about the plants they use and why they chose the plants they use and how they chose them in their practice. And one of the interesting things that emerged was that they were telling her that the use of plants was quite a new thing, and that it seemed that as people's knowledge and awareness of biomedicine, of scientific medicine, was increasing, it became more necessary to sort of to validate a practice by moving it away from something magico-symbolic, using incantation or something of that kind, towards something where you could explain that, well, plants are medicinal and this, I'm using this plant. So the idea of newness in medicinal plants became something that I became very interested in. And we explored that a little bit more, looking at a different method from evolutionary biology, which is where you can perform something called an ancestral reconstruction. And people do this kind of thing all the time with understanding the evolution of plants or other organisms. And they say, okay, so, did the ancestral plant have dehiscent fruits or indehiscent fruits, or did the ancestral plant, was it woody or herbaceous? And these kinds of reconstructions are made by taking a phylogeny and taking the data about the plants and then using statistical methods to infer what the state was, so what the condition was at the time that things branched. And this is a method that can also be used in evolutionary anthropology, and evolutionary anthropologists use this method to infer things about the history of material culture. And what we thought we could do was use these methods to infer something about not the history of material culture, but the history of knowledge of plants. And we chose as a case study the use of plants for medicine across Polynesia. So the Polynesian sort of occupation, the peopling of Polynesia is one of this, of course, really well known, remarkable history of journeying. And we know quite a bit about the relatedness of peoples who live in different parts of Polynesia. We know that, and about their languages, and evolutionary anthropologists have often have made several case studies of the peopling of Polynesia as a way of understanding how people's culture changes through migration and through time. And we decided to bring those methods to bear on this question of, well, how much, how does plant knowledge for medicine persist? So what this is a phylogeny here of the peoples, of some of the peoples. It's quite a small study and we're extending this now. But these are people speaking different languages, language groups, cultures of Polynesia. And this POC is the Proto-Oceanic peoples. So this is a recognized, if you like, ancestral node of the peoples who occupy Polynesia. We found 100 plants that were recorded as used in medicine that were that we could score quite rich data from the accounts of ethnobotanists. And we did that to reconstruct which plants were used in which ways by Proto-Oceanic peoples. And to do that, we collected data about the names of the plants. And if names are similar cross-culturally, then linguists, evolutionary anthropologists call them cognate names. So we looked for cognate names. We looked for the part of the plant that was used and for how the plant was used. So that's the therapeutic application. And we looked at a range, I think about 14 different therapeutic applications of the plants that included in respiratory medicine and in for digestive complaints. And here we see the results of our reconstruction. We found of the 100 plants, 22 reconstructed as plants of putatively medicinal plants of Proto-Oceanic cultures. If you include the therapeutic application and the plant part in your definition of likely continuity of use in medicine, so being used for the same thing and being used the same part of the plant, then there are 4 plant species that we found that reconstructed to this node because they shared therapeutic application and plant parts. And of those, 2 of them had cognate names. So this is also challenges, if you like, the view of stasis of knowledge of medicinal plants, because what we're seeing here is actually a really rapid adaptation as peoples have moved across the Pacific, we've got new plants being used to meet health needs, and from our analyses, less evidence of the maintenance of really sort of like deeply ancestral knowledge. So people are amazingly adaptive, and that's very exciting to me because it challenges some of these ideas. I've got some papers and some work that I'd like to introduce to you that looks a little bit less into the evolutionary anthropology and the way that cultures have diverged or chosen or similar or different plants for their medicinal floras, into some other work that I've been doing, which is more directed towards understanding how the way that plants are used within a culture might give us some clues about where the most efficacious plants are. And this is a study that I did with Esteban Sosa, who's a Brazilian student. He was interested in the use of medicinal plants in Brazil and how their use related to how they'd been studied, and how that in turn related to whether plants were the source of pharmaceutical drugs. And what we have here is the evolutionary tree of all of the genera that belong to the family Fabaceae, the pea family. And so we started with this known set of relationships, how the plants are related. Remember, if you go right back to the Taxol story and the Systematics Agenda 2000, they were about how plants were related is going to help us find valuable small molecules. And here we are exploring that idea with an explicit phylogenetic tree of all of the legumes. And Esteban had collected a huge amount of data about how these plants are used in Brazil. And so he had something like 1,400 use reports that he'd sourced from 15 herbaria, 105 papers that were describing how plants were used. And we looked at the patterns of use across the tree. And you see these branches here that are in red. Those red branches are branches that are statistically using a statistical method to measure lineages that overrepresent a particular trait. So in this case, we're saying the trait is being used in medicine, and these red branches have got more plants used in medicine than expected. So these are what you might call the important lineages in the evolutionary tree, the important branches, but we called them hot nodes, and that seems to have cottoned on as a thing. Finding parts of the tree that are really rich in ethnomedicinally used species, most people will call those hot nodes now. So we found the hot nodes, and then we also got a data set that described how many of the plants that were used, that had been the source of a drug, a pharmaceutical drug, or that had gone as far as clinical trials, where in the tree those plants were. And this funny table here, which when we designed it, I thought, oh, that's a really good way of explaining what we're doing. But now I find it, I think, oh, is it? But what we show here is there are 16 species which are the source of pharmaceutical drugs amongst these plants of Brazil. If you look at every single species, every genus, all of the diversity of all of the Brazilian legumes, looking for a valuable small molecule, you will look at nearly 3,000 species. You will look at more than 200 genera, and you will, of course, get all 16 of the species. That's if you look at everything. If you look at only the species that are used in ethnomedicine, so the ones nothing to do with what they're related to or not, but just things that have a record of being used, you will end up looking at this many species, and you will find 11 of the plants that have yielded drugs. But we thought, well, the best way to really focus in and really limit the amount of screening you do, but find as many of the important drug yielding plants as possible, would be to look in the hot nodes, because these are the ones that are really rich. These are the ones where our data show are really important. But when you look at those, you have this massive drop off in the number of plants that have yielded drugs, natural products that have become drugs. And this was a huge surprise to us, because we thought this hot node thing would be a really efficient way of finding small molecules. But when we really drilled into what was going on here, we found that we've got genera that lie outside of the hot nodes, where there are few use reports and not very many species used, but the uses are for strong effects. So these are for killing parasites, as abortifacients, for in the case of unwanted pregnancies. So these plants are strong effect plants, and the plants inside the hot nodes, they tended to be used frequently, and many of species within these genera were used. They had diverse therapeutic applications, lots of use reports, but they were plants that were, if you like, of mild effect, so nurturing or supportive medicines. And so this was very interesting to us, because it points towards the idea that you can have subsets of therapeutic applications that are of particular interest, but that most plant use, if you want, is of most of the plants that have been screened. So these are how many people have actually screened from amongst these that are available to screen. If you focus your screening on the frequently used plants, which is the case, you're less likely to find the plants that are of interest in the bioprospecting of novel small molecules that have therapeutic value. So that was an interesting study. I'm looking at the time, and I'm wondering if you would like one more example or two more examples of studies, because I've got too much to say. I'll carry on, but let's see. This is very, very briefly. We picked up the idea of plants of strong effect in the last study. Here's a PPP paper recently out. We expected that the plants that have been described as being used in cancer management would be these plants of strong effect, but we find that evolutionarily they belong to the same lineages as plants used for food and plants used for lots of other diverse uses. So the way that these therapeutic applications are recorded in the field and in the literature hides that many of these plants are probably plants of frequent use and mild effect, that they are supportive plants. So it points towards challenges of using the therapeutic applications that are recorded in ethnobotanic literature for bioprospecting. And indeed, don't worry about this in detail. What we find here is when you compare pairs of cultures, And when you look at different therapeutic applications within cultures, so there's lots and lots and lots of tiny boxes here. When they're green, that tells me that plants that are used for one therapeutic application are also used for another, that there's cross-predictivity. Most plants cross-predict between therapeutic applications, and that makes it difficult to use therapeutic applications to predict very specific uses. I can't resist telling you this next and final story, and I'm really sorry if this is too long. But this is some more recent work that I've been doing, and it's using what we know about the evolution of plants, but in quite a different way. And this is work that was done by that was led by a collaborator of mine, Marco Leonti, and anyone who's an ethnobotanist or an ethnopharmacologist will know his name. He's a very sort of prominent and prolific researcher in ethnobotany, and he's been really long interested in what plants taste like, whether that is predictive of how plants are used. And this work describes an investigation of how taste informs the way plants are used, where taste is a thing about a plant, what you can call a plant trait, how that influences the way plants have been selected and incorporated in medicinal floras. So the work was done based on the work of the Materia Medica, which I'm expecting I've got an audience who are interested in history of plant medicine and will be aware of Dioscorides, who recorded the drugs that were used in Greco-Roman antiquity. And so he wrote a Materia Medica, a guide to the drugs of his time, and that has been the pharmacopoeia that has influenced the development of medicine onwards for probably the next 2000 years. This work was based on a translation by Matthiolus, who made a lot of money out of translating Dioscorides. He did very well out of it. He published it in 1544, and in his lifetime it went into 13 editions, and it was translated in, it was part of the spread of Dioscorides as a source of knowledge about drugs. So Marco and I co-supervised. Marco led really the project, the work of the collection of these data, the work of Peter Staub. And what Peter did was he recorded from Matthioli's translation of Dioscorides the therapeutic applications of all of the plant drugs that were included. And he also convened a tasting panel. He collected the plants of Dioscorides and he convened a tasting panel who tasted all of those plant drugs and recorded their chemosensory qualities, their tastes, as aromatic, salty, astringent, smoky, all of these different ways that these plant drugs tasted. And we had the therapeutic uses from Matthioli's translation of Dioscorides. We had the tastes from a tasting panel, and we used these methods which incorporated phylogenetics, where the phylogenetics was used to correct the data. What that means is if you have 7 plants that are mints and they all taste of menthol, you don't say menthol is linked with you don't use those as 7 points that are supporting a relationship between tasting of menthol and being used if you've got a cold, because they're all so closely related they should count as one data point. So this is phylogenetically corrected investigation of the relationship between taste and use, and we find that the stronger you taste, the more uses you find. The more simple the taste, so the fewer number of tastes, as complexity increases, you have fewer uses. This, which you can't see, but you should go to look at the paper, shows which are the predictions in terms of the pairs. And what was really remarkable about this work was how strong the signal was, how well the taste predicted the use. And there were so many. These are the significant predictors here. So significantly predicting not using for use, or significantly predicting use. And so you can see there's a lot of predictions made. And this is really exciting work because it's directing us towards understanding a little bit more how people, perhaps in different cultures, may be using different cues to identify plants to use in medicine. And my current work, that's building a little bit on this work about taste as a cue for therapeutic use, I'm interested in making a cross-cultural comparison of taste and use, where the tastes that I'm interested in are well, not so much taste, but whether plants are attributed hotness or coldness, which could be cued by their taste, but might not be. But it's linked to the idea of humoral medicine, which is a concept of healing that is, contemporaneous with the work of Dioscorides, where you're too hot, you have a cold plant, you're too cold, you have a hot plant, and disease can be caused by an imbalance in this. This is one axis, there are others, but this one axis. And so we made a quick study. This is a tiny last thing, I promise I'm going to shut up. This is a sneak preview of the work I'm doing at the moment, which shows that the perception of plants as hot or cold is a culture-bound phenomenon. And where there's opportunity for sharing knowledge, as in the pharmacopoeia of two pharmacopoeias of India, the Unani and the Ayurvedic pharmacopoeia of India, then we do find significant similarities in the way that hotness or coldness is attributed. We measure, but when you look at China, which of course there has been opportunity for sharing of knowledge about between China and India in terms of medicine, but they are significantly different in the way that hotness and coldness is attributed to plants. So is this a plant trait that is it's a plant trait that's really culture bound, but then dictates the therapeutic uses of the plants in these cultures? So I've got quite deeply into into using the methods of phylogenetics to actually ask quite different questions about the selection of plants. I'm going to stop because I've run over. I'm so sorry. A summary of everything I've said, and thanks to so many people who are my co-authors, InMeda students. Thanks for listening. I don't know if I do something now, but thank you.
No, that's all right, Julie. Thank you very
Sorry,
much.
I did go right to the wire. You've got no time.
No. Speakers often go over slightly, so you haven't
I
done
did
anything
speak very quickly.
untoward. No, it was truly fascinating, Julie, and that's what's coming in through the chat. They wanted you to carry on. So thank you. And we've got quite a few questions. I loved your reference to student health and safety regarding Scholtish and his
Unbelievable.
peyote tasting. We'd never get away with anything like that today. But yes, food for thought. So I'm going to work through the questions from the where did the first one come in? From the top. Gosh, there's quite a few. So going back to your first slides about you, there's a question from Helen here. Was yew bark used medicinally by indigenous peoples?
Oh, that's an interesting question. I think people have looked at that after the discovery of Taxol. And I'm trying to remember, I know that someone in Edinburgh was looking at that, and they did look at I think almost certainly not. I don't know. I can't remember. I remember being interested in that myself, but I can't remember what I found. But certainly the way it was discovered was by a random screen by the USDA, and I think that there are some uses, but not in cancer treatment.
And anything
That's
in
someone's that you're going to have to look that up, Helen,
yeah
because I
anything
can't remember.
anything in sort of you relatives in among conifers at all, or
How are conifers used as medicinal plants? That's an interesting question.
are they? I don't know. I mean,
Probably less than the flowering plant. I mean,
mm,
the flowering plants have accumulated. They're the ones that have been in this amazing sort of war against herbivores, that they've fought with this huge diversity of secondary metabolites. So it's really the flowering plants that have co-evolved alongside herbivores, and that's why they've got all
yeah,
of these amazing secondary metabolites as sort of defense compounds, and they are so diverse. But when you start looking at outside of the flowering plants, I think you do get a much less and much narrower arsenal of chemistry. So I would say, but yeah,
yeah.
interesting question.
Possibly linked to taste if you're going back to your
Yes, yes,
final
that's right.
final thesis.
Mm,
Um, who'd want to eat a bit of pine? So
yeah,
yeah. So I'm going on to the next question because we've got quite a few to go through.
yeah.
This is from Catherine. Do medicinal plants tend to be overrepresented in folklore stroke mythology compared with related non-medicinal species? And how often does the associated myth or doctrine of signatures end up being descriptive of the eventual ethnomedicinal, pharmacological,
Oh,
or pharmaceutical use of the plant? So there's
that's a very
a
rich question. There's a
couple of
lot in there,
questions
isn't there?
wrapped in there. So,
I think that that little plot I showed, with next to Daniel Moorman's book, um, that was a co-author of that was Brent Berlin, and I think it was Brent Berlin's work where he looked at, was it Brent Berlin? I'd have to double check. But anyway, the doctrine of signatures, a lot of people have written quite extensively about that as being a mnemonic for remembering which plants have value. So um, so many ethnobotanists today would point towards the idea of these being ways that people remember amongst a multitude of species that have uses, how the species could be used. And so the idea that they are part of the selection process is given less credence. Um, that's one of your questions, is how important, what do we think about doctrine of signatures? Your other question was about, are the plants that are attributed a sort of magico-symbolic uses more likely to also have medicinal uses? I'm going to write that down as the next student project I'm going to look at, because that's a really interesting question. I think one thing about trying to tease those things apart, and it's something I've been thinking about, is that often when plants are used in healing, we can't tease apart whether their function is a magico-symbolic one, or whether they contain small molecules that are active against the specific therapeutic, for the therapeutic need. So I think it would be quite hard to score plants as magico-symbolic or medicinal, because there's this big grey area in between.
well, maybe another student project.
Yeah, I'm thinking about that. I think that's a really
Julie.
good question. I mean, I think when we come to that from a sort of UK perspective, we do perhaps put those plants into different piles. Here's a magico-symbolic plant that's, I was taught never bring May into the house because it's bad luck unless it's the 1st of May. So, here's a plant with a magico-symbolic belief about it. And then, here's a plant that's good for indigestion. And those are very different things. But I think that's just one way of looking at the world. And sometimes they're not different things.
Thank you.
Very
Yes, there's some good questions here that will no doubt feed into student projects.
Yeah.
And here's another one from Linniver. Are there ethical frameworks used for this kind of work in terms of working, and I presume, and with respecting the original communities?
There certainly are. So any work that is done with a community is done under the will have consideration of the ethics of that work, and that would include a clear prior informed consent process and a clear sort of access and benefit sharing process. And those are, we follow guidelines that have been developed by the Society for Ethnobiology as far as possible. And so, for example, the work that I pointed to with my student in Morocco, there was a period of consultation about whether participants wanted to participate, and then the access and benefit sharing aspect of that was that that knowledge would be returned to the community in the format that was most useful to them, that they would want, which was in the sort of written format in their own language, so that could be and the many copies of that, so that it could be circulated and available in sort of local meeting places and so on, so it could be recorded. So that's the case where there is ethnobotanical field work with what people call informants, so people who hold the knowledge, sharing the knowledge, and that knowledge becoming recorded. But I think it's quite different when there is this huge body of published literature, and most of my work is using these data that have been collected since, then by anthropologists or ethnobotanists, many decades ago. And the people who shared those data, their descendants are here, but the sharers of that knowledge possibly not. And the Society of Ethnobiology does have guidelines about that work, and that using data that's published, and they just suggest that where published data is used, that as far as possible it is the originators of those knowledge are fully acknowledged.
Yeah, in line with the Nagoya protocol, and all that side.
But once the knowledge is published, it's rather tricky. I mean, so if you collecting use data from a herbarium specimen that was collected in the 1930s, it's important that those data are returned, and there is a discussion about that in the literature at the moment about whether those data should, whether published data should remain in the grey literature rather than being collected and made more widely available.
Yeah, very important issues of today, banning all sorts of collections and collected material.
Yeah, absolutely.
Right, I'm moving on again because you've got lots of questions. I'm not sure we'll be able to cover them all, but here's another one. Do those cultures with fidelity in oral storytelling, record keeping, have more accurate, diverse medicinal flora?
So are you thinking that the way that knowledge is passed sort of horizontally or vertically within communities might mean that you have more or less deeper ancestral knowledge.
I guess so, yeah.
So that's an interesting question. I mean, at the moment, this work that we've done with Polynesia and a colleague, it wasn't with me, but someone else had also made reconstructions. I talked about those sort of evolutionary reconstructions. So you can say how far back do you go in sort of like predating the splitting up of ethno-linguistic language groups. I would say that there are there's two examples of that kind of work that have ever been done. So at the moment when people talk about their knowledge, then that is recognized as ancestral knowledge because it's been passed orally and people identify it in that way, and rightly so, and that's as it should be. It should be, people recognize that as their ancestral knowledge, but these reconstructions are very, very few. So the idea that you could get some kind of measure of the way that knowledge passed in different communities and relate that to that kind of ancestral reconstruction, that would be really far in the future for a future research question, I think. Because we're only just beginning to address these kinds of
Yes, as you've shown this evening, so very well. I'm going to have to wind up shortly, but there's Harry has put in a couple of questions, one after the time limit, but one before. So, first question is: do you think some of the commonly used plants for mild supportive use could be preventative for the development of more acute disease?
And that's a very applied question. And as
Yeah.
you can tell from the kinds of research I've been talking about, I am a very conceptual person. And do I think so? That's a really hard question to answer because it's not, if I did have a view on that, it wouldn't be informed by any of the research I've done because the research I've done isn't really about that. And I'm not in any way testing the efficacy of any of these plant drugs. That's not what I do. I don't screen for pharmacological properties. I don't test to see what phytochemicals are in plants. I'm not running a lab where I'm looking at the activity of different extracts. And so what I'm doing is like very conceptual. It's about ways of seeing how people have chosen to use plants. So I think that question, which is a really important and interesting question, is a question that is almost another field, actually.
Yes. You're pointing the way to other more applied aspects
That's a very good
of
question.
research. And the final question comes from Liam. Was there still a relationship between strong taste and therapeutic use, even when the use was for external things rather than when it was being swallowed?
Yes, that I do know because that was in our
I
study.
thought you might, so that I thought that'd be a good one to
That's
end.
a good one to end on. Yeah. So some of those strong plants are plants that might be used in muscular pain, for example, and those are sometimes used externally. So that it's not all about ingestion.
Okay, well, that's a good
And in
succinct.
fact, we did not screen the data by whether it was ingested or in other what people would call the mode of application. We didn't screen the data by mode of application.
Okay, well, thank you, Julie. That's a nice, concise answer to finish with. So
Thank
thank you so much for a
It's been
wonderful
a
talk
pleasure.
this evening that has generated multitude of questions. Maybe we'll send you the last remaining ones so
Oh,
you
silly questions.
you
I'm
can
fascinated
answer
by
those, and
what
we'll
you're asking.
send the answers out to the kind people that submitted them and stayed to the end. So thank you, Julie, again, and thank you,
Thank you for
everybody.
inviting me. It's
Yeah,
been
thank you all.
nice to do this
Um,
retrospective view, going back to the days
yes,
the Systematics Agenda 2000. Gosh.
Good. And thank you all for attending this evening. And of course, you'll be made aware of the recording of this lecture and how you can get hold of it. Also, do look at the website and check out the next upcoming lectures. The next one is in 2 weeks' time with our good friend Sue Burge, who does tours of the medicinal beds at the Botanic Garden, as well as many other things. So look forward to seeing you all in a couple of weeks, and thank you all for joining us. And thanks again, Julie, for a really scintillating talk this evening. So
Very much.
good way to start. Good night, all. Thank you.
Bye. 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 Bye.